Hello students, good afternoon. Can you hear me? Okay, I'm gonna start a poll for the technical check. Please let me know if my audio and video is clear for you. So I'm launching the poll now. Please respond as soon as possible. And for the people who are watching on YouTube, you can just type your messages in the chat box. So I'll be able to reach out to you. Students, I need to know if my audio and video is clear so that we can continue our classes. Please enter the polls and give your answers. Yes. Okay. Thank you for your response. Okay. So I can end the poll now. So today we are going to talk about principles of inheritance and variation. That is part 3 of your chapter, current going chapter. So before we go, I'm Vinay. I'm going to be handling this topic for today. All right. So before we head on to the first topic, okay, before we head on to the first one, let us solve these questions. Okay. So first question we have is, which of the following crosses would produce a genotype of 1:2:1 in F2 generation they have asked? So please think about this question. I'm going to relaunch a poll for MCQs and please answer as soon as possible, and you have a minute to answer the questions. Take a minute to answer the question.
Hello, Vinay, you are on mute.
Yeah. Now, can you hear me, students? I hope you have figured out the answer by now. Can you please answer your options? Okay. Yeah. Option C. Some of you have given option C. So let us see what is the answer now. I'll end the poll. Okay, fine. So the question is, which of the following crosses would produce a genotype of 1:2:1 ratio in F2 generation, right? So when we talk about monohybrid cross that Mendel did, first he used a tall plant and he crossed with a dwarf, right? So when he used this tall and dwarf plant, first he was homozygous tall and it was crossed with homozygous dwarf. So in the F1 generation, so we had one capital T and one small t. So the phenotype of this was tall, right? So now they're asking 1:2:1 ratio. So that means when we cross F1 generation, when we self-cross F1 generation, what would we get? When we cross capital T and small t and cross with capital T and small t, so we get the answer that is capital T, small t, okay? Sorry, cross with capital T and small t, right? So we get that capital T, capital T, capital T, small t, capital T, small t, and small t and small t. So if we observe this cross in the above cross, we can get that we have one capital T and one capital T, that is tall plant, right? That is homozygous tall. So that we can call it as 1. And then we have capital T, small t, capital T, small t, that is capital T and small t, which is heterozygous tall, that is 2. And then we have small t and small t, which is homozygous dwarf. So now we have the 1:2:1 ratio. So our option C is correct. Okay. So next, going to the top, going to the next question. The crossing of F1 to homozygous recessive plant is called as. So this is a very important topic, right? So we have talked about back crossing, like whenever we have our F1 alleles and then we cross it with the parents, we will understand what type of a genotype it is. So in that we talked about what is test cross, right? And what is outcross. So now what they're talking about, when we cross F1 to homozygous recessive plant, they're talking about. So what is a test is F1. F1 was capital T and small t, isn't it? So after we cross the parent, that is capital T and capital T, if I say that this is the parental generation, and if I cross it with small t and small t, so I get an F1 generation of capital T and small t. So now they're telling if I cross this F1 with recessive, homozygous recessive is our homozygous dwarf. So what do I get here? Capital T, small t, small t, small t, capital T, small t, and small t and small t. So this is a cross that is very helpful for us to understand what is the genotype of a particular in a particular organism, like what kind of a genotype. So why is it necessary? So when we talk about phenotypic and genotypic characters, so when we look at phenotypic characteristics, we can just look in our naked eyes and we can understand how they're expressed, and through that we can know that okay, this is how the expression is. But we don't know. See, even though we have homozygous tall and heterozygous tall, they express the same character, but when the genotypes are completely different. So Mendel used this technique of test cross, okay, to understand the genotype of an individual. So whenever we have an F1 that is crossed with a homozygous recessive plant, and that is called as a test cross. Now, is it clear? Okay. So now we are going for the Mendel's laws of inheritance. So when we talked about Mendel's laws of inheritance, which is going to be today's topic. So Mendel's laws of inheritance basically talked about whenever we have two alleles, okay, expressing for a character, one of the allele will express and other allele will not express. So that is what he said. And from there we derived a law called as law of dominance, right? So he derived a law called as law of dominance, which stated that whenever we have two alleles, one will express and other will not express. So similarly, whenever we have dissimilar alleles also, that is one dominant allele and one recessive allele, when they segregate in the following generation, what do we have? We get that they segregate independently of each other, right? So we talked about law of segregation, law of independent assortment, as well as law of dominance with respect to Mendel's genetics. So now, whenever it comes to homozygous dominance or dominant alleles and recessive alleles, how do one allele become dominant and how do one allele become recessive? So that is our point of understanding. So we call one allele as dominant. So any allele to become a dominant, usually they remain in an unmodified state, that is in a very pure state. So when they are in an unmodified state, so they become a very dominant and always expressing alleles. So whenever there is another allele, alternate allele, for example, in dwarfness, if we take, so this is a modified allele where either because of some of the genes that are being modified or lost or mutated, they become modified alleles and they are called as recessive alleles. Okay. So whenever we have two modified alleles, sorry, unmodified alleles, we call it as homozygous gene pair. Similarly, whenever we have two unmodified alleles, we call it as homologous gene pair, and both modified alleles, when they were together, we call it as homologous gene pair. Whenever we have an unmodified allele and a modified allele, for example, we had capital T and we had small t, isn't it? So we call it as heterozygous gene pair. So Mendel's genetics was talking about a certain set of rules for how the alleles will segregate, how the alleles will express, and what happens if two different alleles are together. For example, if we talk about dihybrid cross, right? So when we talk about dihybrid cross, I'll use this here. So we talked about round and we talked about yellow. So these are two different characteristics. So similarly, when we talked about wrinkled and green, right? So forget about the phenotypic expressions, but when we talk about character, color and the shape of the seed, these are two different alleles. One is for color, other one is for shape. But when we looked at the ratio, that is phenotypic ratio in the F2 generation of the dihybrid cross, we had 9:3:3:1 as the dihybrid ratio, right? As a phenotypic expression. Now for this expression, for us to get, it has to prove that the alleles, irrespective whether they are dominant or whether they are recessive, it doesn't matter for them. They go on with dominant and recessive of mixed alleles. For example, capital R went with, okay, small y, and again the capital Y went with small r, right? So that is how we got 9:3:3:1. So we talked about independent assortments, but there were many cases where the Mendelian genetics were not applicable, or sometimes the Mendelian genetics were not followed in many of the cases. Okay, so those cases we will discuss right now. So that is non-Mendelian inheritance. All right. So when it comes to non-Mendelian inheritance, so there are many cases of non-Mendelian inheritance. When you take monohybrid cross, okay, when you take one character similar to that of your tallness and dwarfness, and when you cross it, you had a specific phenotypic expression and specific genotypic expression, isn't it? But whenever you take incomplete dominance or codominance or multiple allelism or pleiotropic inheritance, or polygenic inheritance or pleiotrophism, in all of these cases, they will not follow any of the Mendelian ratios. So we know that what are Mendelian ratios, right? So when I talk about monohybrid cross phenotypic ratio, you will just tell it as 3:1. If I ask genotypic ratio, you will tell 1:2:1. If any of these experiments conducted at monohybrid or dihybrid crosses do not follow, right, a certain Mendelian ratios, then we call it as non-Mendelian inheritance, or something that they're not following Mendelian patterns of genetics. Is that clear? Can I continue? All right, please put a text if you want me to repeat certain topics so that we can do it again. So we'll move on so that we'll understand better. For the first case of non-Mendelian inheritance, our example, first case is incomplete dominance. So what happens in incomplete dominance? See, whenever two alleles are present, okay, whenever two alleles are present, in a heterozygous condition, what happened? See, for example, when we talk about monohybrid cross of capital T and small t, so this is a heterozygous condition, isn't it? So in heterozygous condition, what was our expression? We got it as tall plant. Okay. So the reason why we got tall plant is because the capital T was completely expressing over small t. It is as simple as that. That means capital T was completely dominating over small t. Imagine, okay, I'm not telling that it happens. I'm telling just imagine if this was dwarf character. Okay, if this was dwarf character, then there would not be no, dominance, right? There, you could not tell that the T is totally dominating on small t. Similarly, in one of the cases that we find, that is intermediate expression we call it as, that we can see in snapdragon flower, okay, or 4 o'clock plant, that is Mirabilis jalapa plant, okay. So when we talk about 4 o'clock plant or Mirabilis jalapa plant, now what happens here is we have two characters, like our tallness and dwarfness, we have two characters when it comes to its flower color. So when it flowers, it produces red color and white color, okay. It produces two types of color, that is red color flowers and white color flowers, all right. So now when we talk about incomplete dominant cross, similar to that of our monohybrid cross, see, we have taken in our parental generation one red flower, which is a dominant flower. That is why we call this alleles as capital R and capital R, you can see here. And we have a white color flower, which is small r and small r, which is a recessive flower. So now when the red is crossed with white, so we have one R from here and one small r from here, and then we cross it together. When we cross it together in the F1 generation, what do we get? We get a capital R and small r. So according to Mendel's monohybrid cross, we should get red flower, right? But here in the F1 generation, the pink flower started appearing. So what does it suggest? It suggests that whenever we have, okay, a capital R and small r, that means whenever the two alleles are together, okay, in a heterozygous condition, now neither of the alleles will dominate completely on each other. Neither R is dominating upon R, small r, nor small r is dominating upon capital R. Okay, so it will repeat again. Just look at here. Now, what happens if we self-cross the F1 generation? That is, you have your capital R, small r, and then you have capital R and small r. And when we cross it, we get one red, two pink, and then one white. Now, this particular Punnett square is very important to understand how this do not follow Mendelian laws of genetics. Now look at here, monohybrid cross, right? Phenotype. When we talk about phenotypic ratio, right? When we talk about phenotypic ratio, what did we get? We call it as 3:1, isn't it? We had 3 tall plants and 1 dwarf plants. Similarly, in the monohybrid cross of your 4 o'clock plant, that is red flower and white flower, look at the ratio. What do we get? We get 1:2:1, right? That is 1 red flower, and then we have 2 pink flowers and 1 white flower. So this proves that it is not following Mendelian laws of genetics. So now what can we confer from this? We can understand that in incomplete dominance, right? In incomplete dominance, we can understand the phenotypic ratio is 1:2:1. So that do not follow Mendelian laws of genetics. And next one, what is incomplete dominance? Whenever 2 alleles are in heterozygous condition, neither of the allele will dominate completely over each other. So they will result in a mixed phenotypic expression like pink color. Okay, fine. So is this understood? Please respond through your chat box if this is understood, or if any doubts, you please let me know so that we can move ahead. Okay, so the next one is our codominance. So the name itself suggests codominance. That means 2 alleles are dominating upon each other. I mean, like equally they're dominating. So one of the excellent examples is our human blood group, right? So that is ABO blood grouping. So when we talk about ABO blood grouping, so whenever we want to donate blood, right? Whenever we want to donate blood for the individuals, directly you won't see that your blood is taken from you and infused into the patients, right? They won't put it like that. You take the blood from one individual and you don't put it to the second individual directly. So they collect your blood and run the samples, like what type of blood you are and what you talk about blood typing. I am A positive or B positive or O positive. You talk about blood typing, and which is very essential for us today in medicine, right? So what is the importance? Like how blood grouping is one of the examples of this non-Mendelian characters we'll study now. See, we group bloods as A, B, O based on the presence of an antigen, right? So we have an antigen present on the plasma membrane. See, if you just imagine that this is the plasma membrane of your red blood cells. On the surface of your plasma membrane of red blood cells, there are a particular type of sugar polymerase. Okay, so that depends. For example, if I talk about A blood group, right? In A blood group, the type of sugar is N-acetyl galactose amine. Okay. So this is a type of sugar that is present in the A antigen. Now, if I talk about B, okay, if I talk about B antigen, look here. If I talk about B antigen, now what can I find here? In B antigen, I have another type of sugar that is called as D-galactose, okay, D-galactose. So now you can understand by the chemical name itself, one is N-acetyl galactosamine, other one is D-galactose. So these are the two types of sugar polymers that are produced on the plasma membrane of the red blood cells. Now we'll talk about the antigen properties. Now, if I tell that I am A blood group, that means that I have A antigen on my plasma membrane. So that means I possess N-acetyl glucosamine. If a person tells that he has B blood group, that means he has a certain sugar that is called as D-galactose sugar on the plasma membrane of his red blood cell. Okay. Now, for every expression that happens in our body or any organisms, there are genes responsible for that, right? So one of the genes that are responsible for producing this character is our I gene. So where can we find this I gene? The I gene is present on the 9th chromosome. So out of 23 pairs of chromosomes, right, or 46 chromosomes, we have our I gene on the 9th chromosome. Okay. So when I look at I gene, I genes appear in different forms of alleles, right? So what are the three different types of alleles we can find? See, I gene can appear in three variants. Those alleles are IA, IB, and I. So these are the three different types of alleles. Now I have told you, whenever there is allele A, it expresses a particular type of sugar, right? Whenever there is allele B, it expresses a certain type of sugar. I've told you. I've never talked about small I, right? This small I I've never talked about. So when it comes to small I, it does not produce any type of sugar. Okay. It does not express at all. Okay. So let us see what happens with respect to this particular I. So now we have three different alleles, okay, which is responsible for one single character. And what is our character? Our character is production of antigen on the plasma membrane of red blood cells. Okay. So let's see what happens with respect to the cross. Now look here. Whenever I give my blood for sampling, okay, whenever I give my blood for sampling, now the doctor takes the blood, right? And what he does, imagine that I am A blood group or B, or this is RH. I'll write it as RH factor, and this is a blood type. Now the blood sample is taken and it is put in three different small vials. Now if I add anti-sera A, for the A blood group, now what will happen? My blood group will clump. Okay, we call it as deglutination. So when it clumps, that shows that I'm A blood group. For the same A blood group, if I add anti-B, anti-sera, now what will happen? There will be no clumping. The blood will not clump together. There is no deglutination. That means that he is not B blood group. And RH factor is RH positive or negative based on how it reacts. Okay. Now the person's blood group will become A. Now you can see here in blood sample B, right? Anti-B is added, no deglutination. Anti here in B blood group, you can see that for anti-B, the B blood group will react. So the person's blood group will be B. Now for AB blood group, you can see that they have deglutinated both for anti-A as well as anti-B. Now we can confirm that a person's blood group is AB. Now I have told you the I antigen is not responsible for production of any sugars. That is why you cannot have any antigens present. So no antiseras will work on O blood group. That is why they do not deglutinate. All right. Excuse me. So this is about how we test the blood of an individual. Now let us talk about our blood groups. Now, if I talk about a person having an A blood group, if a person has to have an A blood group, we know that for every single character, an allele has to be in the form of pairs, right? They have to be in the form of pairs. So if a person has to produce A blood group, then he has to have an allelic pair as IA or IA small I. Now, in this condition, in this heterozygous condition, small I do not produce any antigen, so IA will totally dominate on it. So the person who is having IA and small I will only produce A blood group. Okay. Now, when it comes to B blood group, we have IB and IB and small I. So when it comes to AB blood group, so we have IB. So look here. A blood group and B blood group are almost similar. That means in A blood group, we have IA or IAI. In B blood group, we have IB or IB and small I. But when it comes to AB blood group, right? So both the alleles of IA and IB have to be present. That is when we get AB blood group. Now, this is a very, very important concept of understanding for us right now. Now, we'll come back to this. So let us talk about O blood group. The O blood group can be expressed only when these two recessive alleles are together. That is, small I and small I have to be together so that the O blood group is expressed in an individual. Now, when it comes to A blood group, because of the presence of IA and IB alleles, so the phenotypic expression, when you look at a person's red blood cells, okay, of an AB person, what we find is we can find that both A antigen as well as B antigen are equally expressed on the plasma membrane of the red blood cells. So that is why they react to both anti-sera A as well as anti-sera B. And that is why this AB blood group people, even though you give them A blood group or B blood group or O blood group, they can accept without any problem. They are called as universal acceptors. Okay, fine. Now, let us see what happens. Like, what are the parents? Okay, what should be the parents' blood, alleles to get a particular phenotype? So we have parent 1 here, and we have parent 2 here, and we have several different variants of alleles to get a particular set of blood groups. So our intention is, when can we find AB blood group, right? See here, if a parent A is IA and parent B is, parent 2 is IA, then we have IA and A blood group. And then here, IA and small I, look here. If one of the parent is having, okay, IA, which is a dominant, and then we have a recessive. So even a heterozygous condition also we get A blood group, right? So then we have B, and then we have another B, which is a dominant. So again, we have a B blood group. Again, one dominant and one recessive. So again, we have B blood group. But for a person or an offspring, okay, for an offspring to get AB blood group, look here, parent has to have a IA allele as well as IB allele, so that the IA and IB, when they come together, then we will have AB blood group. Similarly, either one parent has to have IB and then other parent should have IA, and both alleles have to come together, then we have AB blood group. So for O blood group, both the individuals, okay, should have this recessive II, small I. When they come together, we have O blood group. Okay. So this is about the possibilities of different alleles coming together to express in a particular phenotype. Okay, so now I think we can understand even better. Okay, give me a second. Okay, so let us take one of the examples. Okay, so let us imagine we have a father and a mother. So our question that I've asked is, imagine that the father blood group is A and the mother's blood group is B, right? So when we cross, when the father and mother, okay, makes children, what are the possible phenotypes? Okay, or if they tell that, okay, one of the babies are O blood group or AB blood group. So what should be the possible phenotypes of the parents? So this is the question. So I'll tell you once again. So if a father's blood group is A and the mother's blood group is B, okay, we don't know the genotypes of the mother and father, right? But one of the children's genotypes we know, that is either one of the children is AB or one of the children is O, okay? So what would be the genotypes of father and mother? So remember always, for a O allele to get expressed, right? For the O character to get expressed, it always have to come together as small I and small I. So this is the first criteria we have to remember. And next one, whenever we talk about a child, okay, having an AB blood group, right? So that means that the child should have both IA as well as IB, right? So this is one small trick to understand how to construct this alleles. So now we have IA and small I as one parent, we'll take it as. Next we have IB and another small I as another parent. And then we segregate. So let us say that the father's character is IA and small I, and mother's character is IB and small I, right? So IA, IB will go together to give rise to AB blood group. IB and small I will give us IBI, that is our B blood group. And then small I and IA, that is IAI will give us A blood group. And small I will give us O blood group. So the offspring should be what? We have A blood group, we have B blood group, we have AB blood group as well as O blood group, right? So this is how we can understand, okay, through a cross. That is the first cross. So if we take a second cross, imagine that the father is AB blood group and the mother is O blood group. So what are the possible phenotypes that we can expect from this particular cross? So we know that the father is having an IA as well as IB. So we'll put it in one side. If a mother has to express an O blood group, obviously she should have two homozygous alleles, that is small I and small I. Is it right? The small I and small I will put it as, okay, mother's alleles. So when they mate, okay, we will get IA and I, which is A blood group, IA and small I, again B blood group, and then we have IA and small I, that is A blood group, and then IB and I, which is B blood group. See, what are our possible blood groups here? We will get only A blood group as well as B blood group. Okay. So did you understand codominance? Is it understood? You can just put a chat. Okay. If you have any doubts, so that we can answer these questions, so that you can feel better about how the things are going. All right. So I'll wait for your chat or messages to let me know, like if you have not understood any of the particular topic. Okay, we can repeat again. So fine. What about multiple allelism? Okay, which is a third example, right? So our third example, we talked about codominance, right? We talked about codominance, and in codominance we took our example as blood grouping, right? So now here the same example can be used for multiple allelism. So I told you while I was explaining our codominant condition that in our codominance, that is in our blood grouping, we have different types of alleles. One is IA allele, we have IB allele, and then we have I allele. So till now, right, when we talked about monohybrid cross, or whenever we talked about dihybrid cross, or even incomplete dominance, when we talked about, we only saw any two types of alleles, either tall or dwarf or red or pink. Only two types of alleles we have seen. We've never seen another allele controlling a character, right? So whenever we have more than two alleles, okay, trying to control a character, okay, so or govern a character, so this we call it as multiple allelic condition. Okay, so in multiple allelic condition also, see, you cannot directly base this on Mendel's genetics. Why? Because according to Mendel, there are two alleles. Okay, they appear in pairs. Okay, the factors has to be dominant or recessive. This is what Mendel was talking about, the factors, alleles being only two, but there are more than one allele, more than two alleles, right? So this is another example for non-Mendelian genetics, that is multiple allelism. Okay, so in very simple, what we can understand about multiple allelism is, for multiple allelism, we require more than two alleles. That is, a third allele is required, okay, for this condition to fulfill. Okay, example for multiple allelism is our ABO blood group. And now if I take one individual, let us take, I take one individual, okay, from your classroom or in my family, or anybody, or any of my friends. If I take any of them, now if I tell what is your blood group, they will tell A. Okay. So if I tell the partner's blood group or another friend's blood group, they will tell, let us say B. So if I take only two individuals, now I will not be able to understand properly what is the genetics, okay, how the alleles are behaving. So now if I take a group of individuals, let us say that one person is having in your classroom A blood group, other person is also having A blood group, then we have B, AB, and O. So if I take a group of individuals, now I will know that who is homozygous, who is heterozygous, okay, who is codominant and who is recessive, right? So whenever we talk about multiple allelism, the possibility of understanding multiple allelic condition comes from population studies. So we have to take a population to understand how the alleles are expressed and what type of alleles are present for a particular character. So this is about multiple allelism. Is it understood? Guys, I'll give you, okay. I'll give you a second. 1 second. Let me check the YouTube too. Okay. So please let me know if you have any doubts. Okay. So we can continue. Fine. Our fourth example for non-Mendelian inheritance is polygenic inheritance. This is a very brilliant example. That is, one of the examples is human skin color, which is very, very, very noticeable in our day-to-day life, right? See, when we talk about human skin color, it is not at all in our control. So we have so many different continents and so many different countries across the globe. So whenever we see different people, right, they come across in different colors. So some are very, very dark, some are very fair, some are dusky, some are brown, right? So you can see varieties of colors being exhibited in just humans, right? So how there are different shades for human color? So that answer, the answer for that question can come in understanding what is polygenic inheritance. Okay. So another example is your classroom itself, height. So whenever you go for assemblies or whenever you go, in your younger classes, where whenever they used to make you stand, they used to ask you to stand in height wise, right? So you can see that how many variants of height would be there in your own class itself. There will be many people who are like short, very tall, medium height. So we are not of same height and we are not of same color. Okay, nor we are having contrasting, like somebody is black, somebody is white, somebody is tall, somebody is dwarf. It's not like that, right? So we have mixed characters coming up here. So now how this particular trait, okay, how this particular character is expressed in so many variants, and the answer for that is in polygenic inheritance. What is polygenic inheritance? Poly means many, genic means genes. When several genes control a particular character, okay, or an expression of a character, that we call it as polygenic inheritance. Now, basically, the polygenic inheritance is very severely governed by environment. Now, let us take one example. If any of you practice a lot of basketball or volleyball, right? So if you play a lot of basketball and volleyball, now what happens? Gradually, your height grows, right? Your height actually grows. So if you are in very, very tropical, where you have high mountains, okay, and then when there is extreme cold, okay, so there is on purity of water, the water you drink and the water you bathe in, okay, or the amount of sunlight you receive on your skin, they all can determine the amount of fairness in your skin. Or even the food you eat will determine whether you are going to become fair or dark. It is very, very possible, isn't it? So environmental factors actually govern a lot. They influence a lot on this particular characteristics, for example, human skin color or human height. Now when we talk about polygenic inheritance, okay, it's a kind of additive. Okay, what is additive? So if I take, for example, if I take the genes, that is A, B, and C, okay, are the three genes that are responsible for controlling the human skin color, let us say that. So what is human skin color? So usually we have our skin, and we have skin cells, and the skin cells will produce pigments called as melatonin. So based on the amount of melatonin pigment, we become darker. More melatonin, more dark; less melatonin, fairer. So that is how we express our character, right? So now imagine we have three different genes, right? So I told it is additive. What is additive? If how much is A expressing, how much B is expressing, how much C is expressing, all the three expressions will add together to give rise to one phenotype. Okay. So I will explain even more better in the next slide. So let us say that for our skin color, our dominant forms are capital A, capital B, and capital C. And for, which are dominant. Whenever I say dominant, that means that they are responsible for giving rise to what color? Dark color skin. So why dark color skin they are giving? Whenever we talk about expression of character and dominancy of character, right? So that means it is expressing to give what? Pigments, right? It is expressing to give pigments. So whenever we have A, B, C combined together, combines together to give us pigments, okay? So that will result in a particular phenotype. Now, when we talk about recessive type, okay? Recessive type means they do not produce any pigments. So that will give us a light color, okay? So, so for a particular person to have the darkest skin, so how an individual gets a darkest skin, you can see that alleles always are in pairs. So all the dominant alleles, that is capital A, capital A, capital B, capital B, capital C, if all together exist, right? So if all together exist and all of them are in the dominant forms, that means they express more and more of these pigments. Then we have what? The darkest skin color. Similarly, so some of the people are very extremely fair. How are they extremely fair? So we have small a, small b, and small c. So whenever we have all recessive alleles, which do not produce any pigments, they get what? Lightest skin, like him. Okay, so all of us Indians, okay, most of our Indians, I'm not being racist, but most of us, what will happen? We have mixed alleles. We can have three dominant, like something like this: capital A, capital B, small b, capital C, small c. Okay, something like this. Okay, when we have mixed recessive alleles and mixed dominant alleles, based on the overall expression of a particular gene, person's expression matters. So this is another example for non-Mendelian inheritance, that is polygenic inheritance, and the examples are human skin color and human height. All right. And our last slide, that is pleiotropism. So pleiotropism is a classical example. So why I tell it is a classical example is because whenever we have a single gene, okay, and if these genes expression can manipulate or can result into multiple phenotypic expressions, okay, which can result into multiple phenotypic expression, this we call it as pleiotrophic gene. And one of the best examples that we are going to study in the next coming classes while we study about genetic disorders, that is phenylketonuria and sickle cell anemia. Okay, and one more example is star synthesis in pea plant. Okay, whenever we take these examples, now what happens is one gene will result in many of the phenotypic expressions. For example, let us talk about star synthesis gene. So the star synthesis gene in pea plant is responsible for both, okay, the starch quantity as well as the shape of the seed. So these are two phenotypic characteristics, isn't it? So whenever we talk about phenylketonuria, okay, and sickle cell anemia, which are mutations, which are genetic disorders that is found in man or humans. So now what happens because of a gene mutation, these individuals who are suffering from this, for example, a phenylketonuria patient, okay, just because of single gene, okay, controlling the character, it will cause mental retardation, okay, reduction in hair, okay, motor functions are very, very bad, okay, skin pigmentation is poor. Similarly, in sickle cell anemia also, we have several phenotypic characteristics. Okay, for example, low organogenesis, the organs will not be developed properly, okay, and they get tired very, very frequently, and the patients are very anemic because of the low red blood cell count in their body. Okay, so these are some of the examples of pleiotropism. Okay, what is pleiotropism? Single genes. When a single gene, okay, expresses, they will result in multiple phenotypic expressions. Okay, so this is about pleiotrophic gene. Okay. Fine. So you can see here, that is the star synthesis in pea plant, right? So the capital B gene is responsible for more starch grain. Whenever there is more starch grain, that means the seeds are going to be large in size, right? So whenever we have a recessive character, that is small b and small b, then that will result in what? Small sized seeds because of the low starch content. One is the starch synthesis is also affected, and the second one is the size of the seeds are also affected, right? So this is another example for pleiotrophism, that is the starch synthesis in pea plant. Okay. All right, people. So now we have a question. Okay. A character which is expressed in a hybrid is called dominant, recessive, codominant, and epistatic. Okay. I'm going to launch polls now. Okay. Hold on. Give me a second. Please answer your question. Okay. So I hope you can see the poll now. I will just move this here. Okay. A character which is expressed in a hybrid is called as what? Okay. Very nice. So you have answered in less than 30 seconds, which is excellent. Okay. So many of you have answered as A, right? So you have picked as A as an option. So let us see what our question says, right? The character which is expressed in a hybrid, obviously hybrid is you have a capital T and then we have a small t. So whenever we have a hybrid character, that means what character was expressed? The capital T character was expressed, right? So the expression happens, okay, in dominant condition, which is right in your option. So whenever we talk about recessive, okay, so whenever we talk about a recessive character, okay, do you think that the expression happens? Definitely no. So in codominant state, whenever we talk about AB, right, in AB condition, right, in an AB condition, which is again a hybrid, both of these alleles will express together, right? So anyways, you have chosen the right answer. So the option should be 1. Okay. So we have reached, I think we have 10 more minutes. I can continue, right? Okay. Okay, next question is Mendel's last law is. So usually people ask first law. So here you have a question called as last law of genetics. Okay. I'll launch the poll again. Please try to answer this question. Mendel's last law is law of segregation, law of dominance, law of independent assortment, and polygenic inheritance. Okay. Yes. Correct. So you have to answer these questions in less than 30 seconds because the question is not lengthy and the options are very, very simple. So there is nothing to panic here. So like what is the last Mendelian character you chose, right? So remember kids, some of you have answered segregation, right? Some of you have answered segregation and some of you have answered, independent assortment. See, law of segregation we talked about in monohybrid cross, isn't it? We talked about in monohybrid cross. So what is the next particular law we studied about? We studied dihybrid cross. So in dihybrid cross, we talked about law of independent assortment. No, after that we never came across Mendelian laws. So we are talking about non-Mendelian laws immediately after dihybrid cross. So that means our option is C, right? So the people who have answered A, it won't be A. So it is actually C, because read your question, it is last law. Okay, it's not the first law. It's all right, you can try again. Okay, so I will end poll now and thank you for the response. It's been very sportive of you. Okay then. So our part 4 of today's class is chromosomal theory of inheritance. Okay, so fine people, what happened back in the days, we have a very interesting story. Okay. What is the interesting story is Mendel was a versatile genius. So his work, whatever he did, okay, was beyond the time. Okay. The people at that time was not even understanding what Mendel was even trying to tell. He was talking about factors. He was trying to build mathematical tools. Okay. Okay. Like your Punnett squares, right? And he was giving ratios, mathematical ratios, to prove that there are factors, and this is how the inheritance happened from one generation to another generation. So that was first thing, and people did not understand that at all. Okay, his mathematical approach was completely alien, okay, to that society back then. Next one, it did not reach a very vast number of community. A lot of people did not even know that Mendel had a work on this pea plants, and he talked about genes or factors, okay, to explain about genetics. Okay, and moreover, back in the days when we talk about factors, Mendel could not prove it. He can just show it through mathematics. So whenever he was talking about mathematics and he was trying to tell that, see, this is how the characters segregate, and these are the ratios of how they have segregated, and this is kind of universal. So people started disagreeing with it. Why? Because one thing, they cannot see the factors. Second one, there is no proof for the genes at during that time, and there was no microscopy, and people did not understand what is meiosis or what is what. So they did not agree at all. And the final problem was, okay, and the final problem with respect to Mendel, okay, was whenever, take for example, in humans, when we talk about in pea plants, we talk about specifically like tallness and dwarfness, okay, round and wrinkled, yellow and green, like contrasting characteristics we talk about. So when we talk about the same thing, okay, when we talk about the same thing in humans, can we talk about like black, white, tall, dwarf? We cannot take just two contrasting character. So there were like mixed phenotypes in the surroundings, right? There were like lots of varieties in the nature. Okay, so Mendel was not able to explain how this much variation is present in the nature when he could only prove that about contrasting characteristics. Okay, so that is why Mendel's work did not get recognized, and it did not get the credits that it deserved back in the time. But all credits goes to, okay, Hugo de Vries, Correns, and Chamark, von Chamark. So these are the three scientists. Okay, see, in 1900s, what they will do is they will independently work on the Mendelian laws of genetics. Okay, like the monohybrid cross and the dihybrid cross, they will start experimenting on different organisms using the same Mendelian patterns of genetics. For example, take De Vries. De Vries worked on, again, 4 o'clock plant, okay, or evening primrose. He worked on it and he understood about monohybrid cross and law of dominance, okay, and he even understood the law of in incomplete dominance. He understood not law of incomplete dominance, just incomplete dominance. He even understood incomplete dominance. So for them, Mendel was making sense. So once they compiled this data, they republished Mendelian work. So once the Mendelian work was republished, okay, so back then the scientific community was growing way too faster. Okay, so when it was growing way too faster, exactly in 1876, now what happens? Something miraculous happened, that is meiosis. Okay, meiosis was discovered. So why I tell meiosis was discovered at this point is people understood what are cells. Okay, now they were trying to understand there is something being separated, there is something being shared, or there is something being divided, okay, during the cell division. And these are chromosomes, right? So there is something being transferred from one cell to another cell. So when these things started becoming more and more, that is when these things were discovered, now that gave a platform for the further genetics. Okay, so what is the further genetics? Now imagine Mendel could prove only contrasting characteristics, right? Like tallness and dwarfness. He could not talk about intermediate or mixed varieties of characteristics. So that is when we have our chromosomal theory of inheritance. So in our chromosomal theory of inheritance, what happens? It was actually a hypothesis that was put forth by Walter Schulten and Theodore Bouvier in 1902. So these two scientists, what they did was they started understanding that. So what did they tell? They told that we have chromosomes, and chromosomes carry these factors that Mendel mentioned. Okay, the chromosomes are the carriers of these genes, and whenever these chromosomes segregate, okay, so it depends upon the chromosome that carries a particular gene, okay, on what character that is going to be expressed. In simple terms, okay, if we have a cell has a chromosome, on the chromosome we have a DNA, and within the DNA we have our genetic material. So whichever the chromosome is carrying that specific gene, right, it depends upon that chromosome on resulting in an expression. So he told that the segregation of chromosomes are responsible, okay, along with Mendelian principles, okay, for the particular character to get expressed. So this is about chromosomal theory of inheritance. This was the hypothesis that was proved. Now, when it comes to chromosomal theory of inheritance, first we have to understand about chromosomes. Now, when we have chromosomes, chromosomes appear in homologous pair, right? So we have a homologous pair of chromosomes. Now you can see here a homologous pair of chromosomes from mother, and then we have from father. So whenever we have a homologous pair of chromosomes, now what happens is these homologous parents of paternal and maternal, they replicate, right? After you have your cell division, right? We have G1, S and G2 phase. So after the growth one, we have synthesis phase where there is duplication, right? So or replication. So whenever we have a replication, now look at here, this will become sister chromatids. So these are called as sister chromatids. So the sister chromatids, now what happens? They pair, right? So the sister chromatids have paired. Now this will become a homologous pair. All right, homologous pair of chromosomes after replication. Now you can see here, how do they carry a particular character, or how do they transfer, okay, themselves during the cell division. Now we know that one of the important cell division that is responsible for conveying a character, okay, or giving rise to next offsprings is meiosis, right? So meiosis is responsible. Why? Most of the organisms who are sexually reproducing, they undergo meiosis for production of gametes. So now we can see that if we have 46 chromosomes, 23 chromosomes are just separated, and it is in the sperms or it is in the ovum, right, after the meiotic divisions. Now you can see that during the G1 phase, we have homologous chromosomes. Now after S phase, now they have duplicated, right? Now they have become pairs, right? So homologous pair they have become. After meiosis 1, now what happens during the anaphase? During the anaphase, now what happens? Pairing will happen. Okay. During meiosis 2, now you can see here in meiosis 1, the pairing will happen. By the end of meiosis 2, now you can see in the germ cells, these homologous pair, it has separated like this, right? You can see the colors. The cream is going with green and the yellow is going with red. It is separating like this. Now you see here, it is very simple. Now if you see in this example, the blue and blue are going together here. Okay. So this A and A will come, this A and small b will come here, and this A and small b will come here. Again here, the small a and small b will come here, and the small a and small b will come here. One more example I will give you. Look here. In this example is a very brilliant example. So we have meiosis. This is the first possibility and this is the second possibility. Okay. So in meiosis 1, anaphase. Yeah.
Please take your chat book.
Yes, sir. All right, students. I'm sorry that time's run out. So we can revise the topic in the next session. So we'll have to close the session right now. So if any doubts, you can ask in the next session, okay? Or write a message to your concerned teacher. So we'll get back to you, okay? Thank you.